Students, in this video lecture, we will start with the new chapter, Biotechnology and its application. So we will discuss about the use of biotechnology in different sectors, such as agriculture sector, healthcare sector, and some another sector which ultimately benefits to the human being. So first we will talk about the use of biotechnology in agriculture sector. So as we know that there are three ways to increase the crop or food production. First in agrochemical based agriculture means utilization of several chemicals to increase the crop productivity. The second is organic agriculture means we utilize several organic materials or we can say the biofertilizers in this case to increase the crop production or crop yield. And the third is genetically engineered crop based agriculture means we employ the biotechnological tools and techniques to increase the productivity. So as we know that the improved crop varieties, better management practices, and use of agrochemicals such as fertilizer, pesticides, insecticides has increased the crop productivity. But this is not enough for the human being. And there is a large gap between the demand and availability of the crop. So the genetically modified crops are could be the possible solution. An organism with altered or manipulated genes or genomes is called as genetically modified organisms or GMOs. These GMOs could have several different properties. So we will see what are the properties we can create in GM crops, genetically modified crops. So first is abiotic resistant plants. So we can create abiotic resistant crop or plant variety which will grow in adverse conditions such as in cold, drought, salt and heat etc. The second is pest resistant variety and the third is enhanced nutritional value. So we can produce some plants or crop with enhanced nutritional value. And the next is minimum utilization of minerals means we can create some plants or crop varieties which will require a very less amount of nutrients from the soil. And the last is insect resistant plant or insect resistant variety. So first we will discuss about insect resistant crop or plant. So GM plants, genetically modified plant or crops, genes are modified in such a way to kill or stop the growth of insects to prevent the plant or crop loss. And the good example is BT cotton. Now we will see the details of this BT cotton. A bacteria named Bacillus thuringiensis produces a toxin protein which kills the insect of some families like Lepidopterans, and examples are tobacco budworm, armyworm, coleopterans like beetles, dipterans like flies and mosquitoes. In bacteria, when this toxin protein present inside the bacteria, then it remains inactive form, or we can say it will this toxin will be called as protoxin when it will not be functional or it will be in inactive form. But when the alkaline medium will be provided, then what happens? It becomes active. So when it reaches in the gut of insect, then the gut provides an alkaline medium to this toxin protein, and The crystal form of this toxin protein forms active active toxin. Now this active toxin binds to the epithelial cells of midgut of the insect, and it creates several pores, followed by swelling and lysis of the cell of the gut, and it leads to the death of the insect. The toxin protein is encoded by a gene named cry, means cry gene produces a toxin protein. And there are several forms of this protein, like cry first AC, cry second B, which controls the cotton bollworm, and the another variant is cry first B, which controls the cod borer. So genetically engineered cotton has been produced, which is having this gene, and this is called as Bt cotton. When insects feed upon this crop plant and the toxin goes inside the gut of that uh, insect, then it kills the that insect. So when the plant expresses this cry protein, then it becomes the resistant to some insects. So this is the good example of insect resistant plant. Now we will see one technology, biotechnological tool, which is known as RNA interference technology or RNAi technology, or gene silencing method. So what is this RNAi technology? The ultimate aim of this RNAi technology is to stop the expression of a gene, or it kills or stops the viral genome which is having RNA genome. And the genetically engineered recombinant DNA has been prepared in such a way it encodes an RNA which becomes the complementary of the our target mRNA of a gene, or which we want to silence, or either it is the complementary to the viral genome of the viral RNA virus. When since we have created an RNA which is complementary to the gene, then what happens? Both of the strands becomes complementary to each other, so they will hybridize and they will form the double-stranded RNA. And there is a rule that in cytoplasm, if double-stranded RNA is present, then double-stranded RNA is degraded. So the specific gene along with our mRNA, expressed mRNA, will be degraded. Either if we are thinking about if we are targeting a RNA viral genome, then what happens? Our mRNA means the mRNA which we have produced by genetic engineering will bind to that viral genome. Then it becomes again a double-stranded RNA, and again the genome will be degraded. Thereby, silencing of the expression of gene happens, or we can say the translation stops. Or in case of viral genome, when the double-stranded RNA is formed, then the genome of that virus will be degraded. So in this way, we can silence a gene and stop the growth of RNA virus. Now we will see the details of pest resistant crop. So what is the pest resistant crop? Means we modify the genome or some genes in the crop so that it becomes resistant to the pest. And as we know, the nematode is the very harmful to the plant as a pest. So delivery of nematode specific genes into the host plant through agrobacterium vector is done. We have already discussed this uh, vehicle or vector, agrobacterium vector. So we used to utilize this agrobacterium vector, and we utilize the delivery of nematode specific gene into the host of the plant, and this gene encodes a complementary RNA to the nematode RNA, and this nematode RNA should be uh, very essential for their survival. Either we can say, in another word, that this RNA should be involved in the function of vital role of that nematode. So when it becomes the double stranded RNA, then the nematode cannot survive on those plants having such arrangement. So what happens when this nematode infects or uh, inhabits this pest resistant plant, then the expressed mRNA will neutralize the specific gene of nematode, which is very essential for their survival. So the nematode will be killed or they will die ultimately. Thank you.